Good morning, students, and welcome to the online teaching session. Students, today we will take up the new topic XGP, named as Human Genome Project. Okay? Now, see, children, from the name, it is clear that in this section or in this topic, we have to study about the human genome. That is, we have to study the details of the human gene and its composition. So this is the Human Genome Project. And why it is called as a project? Because there were few scientists who have taken the pride, you can say that, who have taken the responsibility to study each and every human gene. Okay, and then only they found its application. That is, if any of the gene is faulty, like we have already studied about some of the genetic disorders. So what the scientists, they found that if any of the gene is faulty, if any of the gene is wrong sequenced, then it can be treated and we are able to cure the genetic disorders also. Okay, but what are the conditions for this? This we will see afterwards. But you remember that in case of Human Genome Project, we studied the composition of each gene. Okay, and let us begin this Human Genome Project. Now see, children, this Human Genome Project is also called as a mega project. Okay, so first we will see some points in case of HGP. It is called as a mega project. Now, why it is called as a mega project? Because it was estimated that there are 3 into 10 to the power 9 base pairs are left to be sequenced. Okay, so what was the estimated? The estimation was that there are 3 into 10 to the power 9 base pairs are present to be sequenced. Okay, now understand the meaning of 3 into 10 to the power 9. That's why it is called as the mega project. Okay, it means the genes which is present in our body, it has to be sequenced properly. Okay, it has to be studied properly. And if there is any fault, there is any mistake, then that can be corrected. So when we can correct it, if we know about the human genome sequence, if we will know all these things, then only if there will be any wrong thing or any faulty thing, then that can be cleared in the patient. Okay. So, what we have to study? That there are 3 to 10 to the power 9 base pairs are present. Now, next point. The cost of sequencing one base pair was $3. Clear? So, what is the cost of sequencing per base pair was assumed, it was just the assumption, was $3. Okay, and the cost of total base pair, okay, cost of total base pair became 9 billion. That's why it is called as the mega project, because it requires a huge amount of money, clear, as well as many numbers of gene was there to be sequenced. Then, see one more point. The point is, at that time, when these genes were going to be sequenced, we have no any specific place to collect the data. Like nowadays we have the computer system and everything. So now we are able to collect each and everything in the form of a data, like we can store. But at that time there was no any storing device, and so a new branch of biology was introduced, and that is called as the bioinformatics. So what was the next point? That is, due to lack of storing data, a new branch of bio was developed, okay, or introduced. Because in case of biology, till that time we were only studying about the zoology, microbiology, botany and all these terms. Okay, we were not studying about the biotechnology. Actually, all it comes under the biotechnology. Clear? So, what we have introduced? We introduced a new branch of biology that is called as bioinformatics. Clear? Next point. Now, while sequencing this gene, there should be some ethical, legal and social issues. Definitely, we have to take permission that we are going to study the human genome. Okay, we are going to study about the genes of the human being, and if something will be faulty, then it can be corrected. So there should be, or there was some legal, ethical and social issues. So to avoid this, okay, to solve the problem of all these issues, a committee was established. So for this reason also we can say that this XGP was a mega project, because for the first time, to resolve the problem of ethical issues, social issues, okay, and legal issues, one committee was established. Clear? Next point. It was estimated that this project would be completed in 13 years. And it was started in 1991. And so, according to the 13 years, the scientists thought that it would be completed in 2003. But actually, it took 3 extra years. It was completed in 2006. That we will see afterwards. Okay. Now see. All these are the points. The next and important point is, this project was started by, okay, US. So what to write? Our ninth point will be, it was started by US Department of Energy. Okay. US Department of Energy, okay, and National Institute of Human Health. National Institute of Human Health. Okay. So, this is the concept of the HGP. That first of all, what had happened? Some scientists, they must have gathered together. They have decided, or they must have decided that what they had to do. So they thought that we'll study on the Human Genome Project. Okay. Now for the Human Genome Project, they have sequenced out the human genes, and they found that there are 3 into 10 to the power 9 base pairs. Okay, and then they thought that if sequencing one genome will cost $3, then there will be altogether $9 around the $9. And after that, now again one problem came in front of them, that is where to store the data. So a new branch of biology was developed, and that is called as the bioinformatics. After that, what other problems were there in front of them? There were the social, ethical and legal issues. Just to resolve those issues, they have developed one committee. Clear? So before starting the project, they all had prepared a lot. Okay, and then it was estimated that the project would take near about 13 years. Okay, and as it was started in 1991, it was assumed that it would be completed in 2003. So first of all, the US Department of Energy and National Institute of Human Health, they have started this project. Okay, now we will see what is the concept of the project. What is the concept of the Human Genome Project? Actually, there were the two groups of scientists. Clear? So, one of the groups was telling that if we are coding for something, then why should we go for the non-coding part? Clear? And another group of scientists, what they were suggesting, that we can code the whole DNA instead of the coding and the non-coding part. I have already taught you children, when the DNA gets converted into mRNA, then this DNA strand first is changes into the exon and intron. Then exon is the coding part, intron is the non-coding part, and exon will be sequenced together, and then it will form the mRNA. Then capping and tailing would be there. Okay, so this I have already taught you. It means the two group of scientists were there, and some of them were favoring that we can code for both, and another group was favoring that we can code for only coding part instead of the non-coding part. So in this way, the two approaches came forward, which gave us the methodology of the HGP. Now we will see the methodology of the HGP. Okay, now see. So two approach came. Okay, for the Human Genome Project, two approaches came. What was the first approach? Like I can write two approach. So the scientists worked on two approach. Number one, to sequence, okay, only coding part. And next, to sequence the whole DNA. This is called as expressed sequence tags. EST. Clear? And the second one is called as sequence annotation. Okay? So this is the main approach by which we have to sequence the DNA. Clear? Now, next point. Second important point. It was important thing. Now, what to do in case of methodology? Now, see children, this you will better understand when we will start the biotechnology. But I am giving you the method of taking out the DNA. So, what is the process of DNA? With the taking out the DNA, with the help of some of the enzymes. Okay, now enzymes would be any. Would be any means it can be ligase, it can be protease. Okay, so we will see. So on the basis of the enzymes, what we have to do? We have to remove the cell membrane. Clear? And after removing the cell membrane, we'll insert inside the nuclear membrane and we'll take out the DNA by using an enzyme named as endonuclease. Now this enzyme would cut the DNA into the pieces, and then by the various steps we will sequence those DNAs. Okay, and then we would get the desired DNA. Now what we will do in all these cases, like suppose we have used, now see what terms I have used. First, what to do? We will use enzyme. Okay, now along with this enzyme would be ligase, nuclease and protease. Okay, all these enzymes are there. With the help of these enzymes, we will take out the DNA. And by using one more enzyme that is called as endonuclease, will cut the DNA. Clear? Then we would take the desired piece of DNA. Suppose this is a long strand of DNA and I have simply made one cut here, and this is my desired DNA. Now this DNA will be introduced into the body. Clear? So before that, how we can introduce this DNA into the other body? We can insert this with the help of vectors. So your next topic will be vectors, and then in which we are going to introduce, clear, host cell. So we need vectors, we need host cell, we need important enzyme endonuclease and some more enzymes. Clear? Just to cut the DNA piece, take that small piece of DNA, because we have to sequence this, and put the sequence in the vectors, and after that we'll introduce it into the host cell. Okay. Now we have used two important vectors here. One is BAC, bacterial artificial chromosome, and another is YAC, yeast artificial chromosome. But after getting all the informations and everything, we came to the conclusion that this YAC can be treated more efficiently. Okay, so by using the vectors, by using the host cell, by using the enzymes and endonuclease, we can prepare, we can sequence the gene. Okay, and that can be introduced if it is required. Now, what to do? We can make multiple copies of the DNA by introducing it into the host cell through the vectors. Again, this you will understand when we will continue with the biotechnology. That suppose children, if somebody is having a problem of diabetes, okay, then we used to give him the insulin either in the form of injections or some other way. So how we can prepare, how we have prepared this insulin? Because our body is capable of producing only that much of insulin which is required for our health. So from where all these insulin have come in the market? Have you ever thought about this? No. I am sure you must have not thought about this even, that from where the insulin bottles are coming into the market. Okay, although our body prepares, we have already studied this thing in the previous class, that our beta cells, Langerhans of beta cells, they used to prepare the insulin and everything, and this is introduced into the body, okay, to those who are the sufferers. So how we get this insulin? This is the question. Now what the scientists have done, they have taken the gene of insulin, okay, from the body. Clear? So see, this is the concept of Human Genome Project, and this is the introduction of biotechnology also, children. So what the scientists have done, they have taken the gene of insulin, clear? Then they have inserted this gene into the host cell with the help of the vectors. Clear? We'll see this thing in detail. Now, as soon as the host cell multiplied or replicated, the gene is also get replicated. Then what the scientists have done, they have killed all these host cells, okay, taken out all these insulin. Then They have purified it, packed it, and then sailed it into the market. So this is the basic concept of Human Genome Project, that we can treat with the diseases. Clear? So now what happens? If the fragments are replicated, clear? Then this sequence of replication was done by the scientist named as Frederick Sanger's method. So method of replication of these gene, okay, into the host cell is done by the Frederick Sanger's, and that's why it is called as the Frederick Sanger's method. Clear? So this is all about the methodology of the Human Genome Project. Now we will see its salient features and the application of the Human Genome Project. Okay, so understood what to do, why it is called as the mega project. We have seen what is the methodology, what we adopted. We adopted that there are two types of gene sequence, coding as well as the non-coding. If we are using whole, then that is called as the sequence and notation. And then we adopted the methodology. Now what we have done in the methodology, we simply cut the cell membrane with the help of the enzyme. Then we cut the DNA with the help of an important enzyme named as the endonuclease. After that, we took the fragment of the DNA and introduced that DNA into the vector. And as soon as the DNA was introduced into the vector, this vector is allowed to introduce into the host cell. Now here the host cell can be anything. It can be bacteria. Generally we used to take bacteria. The detail we will see in the biotech. Don't worry, okay? So what to do? We have introduced everything into the host cell, and as soon as the host cell gets replicated, okay, our desired gene was also replicated. Then take out everything, sequence everything, okay, and then insert it into the, or pack it into the bottles for the further use. Clear? Now we will see what is the salient feature of this Human Genome Project. Okay. Now that will continue in the next class. Thank you.